So thank you very much for invitation. Welcome everybody. Thanks to organizer for the invitation. Today life science, as we know, for over 100 years has proven its weakness in fighting many illnesses and fighting the current pandemic. In 3 months, we went from working on the genome improvement to the medieval solution, stay at home. We should realize that it is necessary to heal the cells and not modify them. The current market does not have any therapy to heal, and it is a necessity to introduce a new solution. And I will tell you about cell therapy by elimination of the chemically modified protein using MEMS. The fatality of current pandemic is not the virus, but the lack of respect of the mechanism of the disease. So drug development nowadays is a lottery. With MEMS, a new technology, we heal the cells which can fulfill their role again in protecting themselves and the body from any infection. I will explain to you the pathology of infection, the role of our new technology of MEMS, and why we must put on the market without any more delay. So, as you know very well, the young people are healthy, but they have infection, trauma, and they became ill with aging. In the last 30 years, the researchers studied the genetic theory of the disease development based on healing of transgenic mice. The people don't become transgenic with age, but their protein is modified by chemical. This protein is not degraded and remain in the organ, leading to loss of immune recognition, cell degeneration, and inflammation. The major chemical which is formed during infection is xanthorrhenic acid. And here you have the western of wild type cells and cells transgenic in the apoptotic protein bit and double transgenic back. So as you see, the protein-protein interaction is completely different. And when we would like to use the drug for white cell type for the people, we cannot switch from a transgenic system because the results are completely different. So now we have COVID-19 induced pathology. COVID-19 leads to pathology because the oxidative stress causes the formation of endogenous xanthorrhenic acid, which modify the proteins. The modified protein lead to the organ failure and death or pathology after infection. COVID-19 leads to post-infection pathology in children and the young people. The cell death is called apoptosis, and it is a regulated process linking to the tissue homeostasis and tissue renewal. Once the protein became covalently bound, we have the protein, pro-apoptotic protein bags. We have translocation of this covalently modified protein into mitochondria and destruction of mitochondria. We have destruction of mitochondria. And if we have destruction of mitochondria, the process is non-reversible. We have the oxidative stress, and we have the problem, respiratory problem, in the COVID-19. So the process is irreversible once we have the modified protein. So modified protein lead to cytochrome C release. It activates the caspase 9, caspase 3, and lead to DNA disintegration. But the protein, which is modified by xanthorrhenic acid and not degraded. So we have such a situation that we have a ghost cells, and these cells and only the fiber in these cells, we don't have DNA, and the cells lead to pathological apoptosis of the surrounding cells. The chemical modification occurs on the secondary amino group of the amino acid of the protein, leading to their later polymerization of the modified sequences. So the chemical modify our protein in the stable, covalent, and irreversible way. The polymer occlude the receptor in the cell membrane. The membrane is misfolded, and the protein-protein interaction is incorrect, leading to future activation of caspases and the cell degeneration. The protein polymer in the cell membrane is the upstream cause of the cell pathology. They lead to caspases activation in cell degeneration. We have new way to remove this protein. We call them molecule establishing membrane signaling, MEMS, which target the modified protein in the membrane. MEMS is the first and unique treatment of the cell degeneration in aging and infection. Molecule establishing membrane signaling binds to polymer on the receptor. MEMS detach the polymer, and the liberated receptor became correctly folded. Polymer bound to MEMS bounded to MEMS is recognized as an antigen. A correctly folded receptor eliminates the antigen systemically. Pathology is stopped. Here we have the example how MHC1 receptor is modified by xanthorrhenic acid. This receptor cannot work anymore. In presence of MEMS, on the bottom picture, the receptor is stopped, the receptor is clean. In the COVID-19, we have severe damage of the cardiovascular system. And we see here on the upper photo that actin in the presence of modified protein is cleaved, is the actin disorder, and this process is irreversible. What happened in the pathology of COVID-19? When we have MEMS, the cell, the homeostasis of cell is restored. So if we have such a system, degraded cells, as on the left of left picture, we have of course a pathology. And what happened in COVID? We have cell death, which is irreversible, which is caused by the chemically modified protein, and we give to these people chemicals. So these people will die. Here we have on the left side, we have homeostasis of the cell restored in the presence of MEMS. In COVID-19, we have pathology in the brain, in the whole body. And in the presence of xanthorrhenic acid, we have aggregation of beta amyloid. And this is only prevented by MEMS. So the MEMS removes the primary cause of the pathology. Modification of the cell's protein by chemicals lead to cell death. The people taking chemical for their metabolic disease have the modified protein which increase with the COVID-19 infection, and the people die. MEMS enable the host to remove the modified protein. MEMS protects against infection, heal, and allows rapid recovery. MEMS is antibody against the polymer of the chemically modified protein, purified by affinity chromatography. It is not toxic. It is not mutagenic. MEMS restore the immunity in the cyclophosphamide mice model of immunosuppression. The MEMS heals the infection with Staphylococcus aureus in mouse.
Halina,
the basis of the cell pathology is the modification of protein by chemicals. And we would like to hear that chemicals lead to covalent modification of protein. And when we have the covalent modification of protein, we have this modification in the wild type of the cells, and in the transgenic cells, this modification is completely different. We cannot switch from one system to another. And this I tell you that in COVID, we see the pathology in all body, which is caused by oxidative stress, and xanthonenic acid, which is produced and modified all protein in whole system. We can stop only this pathology if we remove the modified protein. So the process which is regulated, the process of apoptosis, is became non-regulated, the degeneration progressed, and it cannot be stopped. The mitochondria destroyed, the cytochrome C from mitochondria is released, and we have the permanent apoptosis, pathological apoptosis. So this apoptosis is pathologic because the DNA is degraded, but the protein is not degraded. So we have such a situation. We have ghost cells which are full of fiber, but they don't have the DNA. And what happened in all this is associated with aging and in all infection. And this modification of protein occur on secondary amino group of amino acid and lead to later polymerization. The polymer is stable, they remain in the cells and remain in our body, and we cannot remove them without our MEMS. So we have the situation that polymer in the cell membrane is folded, and protein interaction incorrect, leading to future activation of caspases and cell degeneration. And we have now the situation that polymer remain in the membrane, lead to the cell pathology. And this pathology cannot be stopped by any antibody, by any chemical. It leads to the caspase activation and cell degeneration. So we have the new technology, molecule establishing membrane signaling, which target the modified protein in the membrane. The MEMS is the first unique treatment of the cell degeneration in aging and infection. So molecule establishing membrane signaling binds to polymer on the receptor. MEMS attached to the polymer, and the liberated receptor became correctly folded. Polymer bounded to MEMS is recognized as antigen. A correctly folded receptor eliminates the antigen systemically. pathology is stopped. Here some examples. Here we have MHC1 receptor, which is modified in presence of MEMS. The receptor is clean. Here with COVID, we have all type of pathology. We have cardiovascular pathology because actin is cleaved, and we cannot remove this cleavage of actin if we give more chemicals, more cleavage. On the bottom photo, we have homeostasis of the cell, which is maintained, and the cell life. Here is if you have such cells, like on the left, the red is the calcium. The cell is completely degraded, and the process is irreversible. With MEMS, we keep the homeostasis of the cells. So in COVID, we have here that we have a brain problem, we have neuro degradation, so we have beta amyloid aggregation in presence of xanthorrhenic acid, in presence of every infection, and especially with COVID. So when we use MEMS, the aggregation not occur. Modification of the cell protein by chemical lead to cell death. The people taking chemical for dire metabolic disease have the modified protein which increase with COVID-19 infection, and the people die. MEMS enable the host to remove the modified protein. MEMS protect against infection, heals, and allows the rapid recovery. So MEMS is the antibody against the modified small sequence of protein polymerized together, and purified antibodies purified by affinity chromatography. MEMS is non-toxic, is non-mutagenic. MEMS restore the immunity in the cyclophosphamide mice model of immunosuppression. The MEMS heals the infection with Staphylococcus aureus in mice. So a competitive advantage of our Axanton technology, MEMS is global anti-degenerative molecule, safe and efficient. It is signaling molecule active at 5 micrograms in water, used sublingually once a week. MEMS is efficient in human against infection for 13 years. MEMS is not a drug, nor a device, nor a food complement. Thereby, MEMS needs to be considered as a whole new technology. The new technology is patented for prevention and treatment of cell degeneration in US, Japan, Canada, and EU. So in COVID pandemic, we have chemically modified protein of the cell membrane, which cause lack of virus recognition. Current market does not have any therapy removing the chemically modified protein. The COVID-19 pandemic exited urgent marketing of MEMS. In conclusion, chemically modified protein lead to cell death. That cell degeneration is upstream event in the pathology in aging and infection. Elimination of the chemically modified protein by MEMS is the unique method of prevention and early treatment of degenerative pathology, COVID-19 and all infection and degenerative disease. And MEMS is ready, active, and safe to protect against the pandemic. Thank you. Thank you for your attention. Okay.
Here we go. Are there any questions from the audience?
Okay. No question.
Yeah, thank you very much, Elaine.
Thank you.